I'm Zachary Cartwright. This is Water in Food.
When it comes to something like rice drying, you're dealing with movement of moisture from within the rice to the outside environment.
This area of research holds huge promise, no doubt, in making our foods more safe.
There's great need to come up with techniques that not only can optimize the yield, but can also increase the value of rice that is harvested.
Welcome back to another episode of Water in Food. Today I'm joined by Dr. Griffiths Atungulu and his graduate student Abass Oduola to discuss the rice processing program at the University of Arkansas. Dr. Atungulu's background is in agricultural engineering, while Abass is working towards a PhD in cellular and molecular biology. Let's hear what they have to say on water and food. Thank you both for being here today, and happy Friday. How are you guys doing?
Well, we are doing very well. Thank you very much for having us.
Of course. And Abass, what about you? How are you?
I'm very well. Thank you so much for this.
Of course. I've been hoping to get some university members on the podcast for a while, and I'm happy to have you guys as the first podcast guest coming from the University of Arkansas. Can you tell me how long you've been at the University of Arkansas, Dr. Atungulu, and what brought you to this university? How did you end up there?
Oh, thank you. Thank you very much for the opportunity. Yes, I've been at the University of Arkansas for close to 8 years now, almost going 9. And before moving here, I was at the University of California, Davis. And at the University of Arkansas, my area of expertise is primarily in grain processing and post-harvest systems engineering, and I'm a professor here. At the same time, I'm also the interim director of our University of Arkansas rice processing program. And so, like I mentioned, my area is in grains, and Arkansas is one of the grain hubs of the United States. We have a lot of rice. In fact, produce 50% of the U.S. rice. And so, conducting research in this place is a very, very good area for my kind of training. And so, that's what I do. Conduct research and also teach at the same time.
And you guys are just starting your semester now. What's that look like for you? Are you teaching in person? Are you back online? What kind of system do you guys have for this upcoming semester?
Well, we have all types of systems going on here, really. We have primarily face-to-face. We decided to come back to face-to-face, but of course, we are giving allowances for a few cases where we may have to implement hybrid systems. There's good flexibility and good coordination from the administration for us to offer students a learning environment that will provide them with the highest level of education, but at the same time maintain the highest level of safety where their health is concerned.
And Abass, what about you? How long have you been at the University of Arkansas and working on your PhD?
Thank you. First off, I'm very happy to be here today. And I've been working on my PhD for about 3 years now. I joined Dr. Atungulu's lab in the fall of 2018. So I'm just starting my 4th year of my PhD program.
Well, congrats. Is the end in sight for you? How much more do you have for your PhD?
Well, it's hard to tell for every PhD program, but I believe with hard work and hope, the end is in sight.
Mm-hmm. And what brought you to this specific university? How did you find Dr. Atungulu's lab, and what excited you about working with him?
Well, before I moved to the United States in the fall of 2018, so I am originally from Nigeria. So I was searching for schools in the US. My keywords were the research I'm interested in. And Dr. Atungulu's lab came up, and I reached out to him, and we had several meetings. So after a while, I realized that what he's doing is a perfect fit for my current and future goals. And to add to that, Fayetteville is ranked the 4th best place to live in America.
Mm-hmm.
And again, we have a lot of rice in Arkansas, so it's a win-win situation for me.
Well, perfect. And Dr. Atungulu, I understand that you are working with the rice processing program. What is this program and what's the goal of it?
Thank you very much. So maybe just a brief history. I can't mention the rice processing program without really mentioning our founder and the first director of the program, who was Terry Seven Morgan. Terry Seven Morgan conducted a lot of research in the area of rice post-harvest processing, and in 1994, he established the University of Arkansas rice processing program. And the primary goal really was to conduct research in the areas of pre-harvest physiology, drying of rice, milling, storage, end-use processing, and sensory analysis that would address the needs of the U.S. industry. And the ultimate goal of this basically was to enhance the quality and the value of rice and rice products. And as we've mentioned, we have a lot of rice. We produce most of the rice in the U.S. We have different cultivars of rice, and you can agree with me that there's great need to come up with techniques that not only can optimize the yield, but can also increase the value of rice that is harvested, preserve it from deterioration, and make sure that the producer gets the highest returns out of the commodity. And so that's what we do. Uh, After its establishment, industries have come on board and become sponsors of the program. We have several industries that sponsor the program. And what they do is they give us feedback on what are the research needs, and we conduct research in coordination with our students. And then at the end of the day, we release those results back to them so that they can implement them to improve their processing. I would say that every year in May, we hold an annual meeting at which our students provide feedback in terms of presentations, and we also get some feedback from the audience. And so that is the nature of the program that has grown to become very impactful. I think one of its kind in the US, and maybe one of the leading rice processing programs in the world.
And Abass, how are you contributing to this program? What is your research focused on, and how are you giving back to this program?
Thank you so much for that. Dr. Atungulu has kind of mentioned about the diversity and different areas of rice research in the West of Arkansas. Well, as far as my research is concerned, I'm focused on the microbial safety of rice. Talking about bacteria and fungi. So my research is actually focused on narrowing down to mycotoxin contamination of rice. So we use different novel equipment such as microwave and infrared to dry and disinfect rice so that our rice will be free of harmful fungi and toxin.
And how are you conducting your research? And maybe you both can answer this question. Are you doing research in the lab? Are you partnering with farmers in the area? What does that look like?
Yeah, so if I may jump into that, there are almost three ways that we conduct research. Number one, like I mentioned, we are in an engineering area, and we do a lot of modeling and simulation of situations that cannot necessarily be uh, done experimentally. And so we have a lot of computational mathematical models that we develop and build. Most of them are around heat and mass transport. When it comes to something like rice drying, you're dealing with movement of moisture from within the rice to the outside environment. And so it's very important. Sometimes we do a lot of modeling, how that rice moves from the center of the rice all the way outside. It's very hard to do a measurement. And so mathematical modeling becomes very, very important in those kind of diffusion type of research. Once we do those models, of course, we do validation. In terms of validation, we definitely have to set up some experiments in the lab that capture the design that we try to model, and then validate those models. And typically, after we do those validation, which are pretty much experimental in the lab, the goal is to implement some of these techniques or the results in the industry. And so we have to coordinate or liaise with the industry partners and see how we can implement some of these or test them on a larger scale. Some of this might involve equipment development. It might start from maybe just designing on the computer to building it in the lab, and then finally trying to implement it at a higher scale to meet the kind of throughputs or space footprint requirements, environmental footprint requirement that the industry might be needing. So I would say we address research in 3 stages. Number one, we do mathematical modeling. We do lab-based research to prove some of those mathematical models. And then we also try to scale up these techniques working in conjunction with the industry partners.
So you're doing some modeling, some lab research, as well as some field trials. Is that correct?
Excellent, yes.
And you briefly mentioned moisture there. And Abass, I'm wondering if you are using moisture in your research and if you're using water activity as a measurement. And if so, how are you taking those water activity measurements and why are they important?
Thank you. So in any microbial work, moisture and water activities are very important to determine the growth rate of microorganisms on food matrices. So for sure, moisture and water activity are very important for me. So we have an equipment called vapor sorption analyzer, simply put, VSA, that we acquire from your company, Aqualab. And this equipment has been of great use in determining the moisture content and water activity and how we can use that to limit or address microbial growth on rice.
So you're using the Aqualab VSA to create isotherms, and then you're using that to predict if microbial growth can occur. Is that correct?
Right. So that kind of gives us an indication of what could happen over time. So like my advisor mentioned, Dr. Atungulu mentioned, we still need to do some storage to actually see experimentally what is going on over time. And also, I also heard in one of your previous podcasts about the Aqualab 40E equipment, and I kind of mentioned about the academic discounts. And I think that is what we, at some point, we may want to explore the opportunity.
Yeah, Meter Group has definitely made it an effort or a point to work with universities like you guys and lots of other universities because I think it is important for you guys to have the best tools that are out there so that you can measure water activity or create isotherms. And a lot of what you do drives the industry and they see what you're doing. And so we're always happy to partner with universities like you guys. Dr. Atungulu, what else do you hope to do with these isotherms? Are you looking at shelf life? Are you looking at physical properties of the rice? What is your focus with this type of data?
Like I mentioned in the beginning, when you come to processing of rice, rice is harvested at about 21% moisture content to 23% moisture content. Ideally, we need to remove moisture out of that rice and bring it down to about 12.5% or 13% moisture content. When you are removing this moisture, the rate at which this moisture is removed is very, very critical to the integrity of the rice kernel. And the temperature at which you're removing it is also very, very critical to the material state gradients within the rice kernel. As you know, rice is composed primarily of starch, and as the moisture reduces, the material changes from one state to the other. And those transitions are very, very important to pick when you're doing your drying process, because if you don't do it right, there's some things we call fissures, or in general terms, we call them small cracks. Those cracks, when they are established within the rice kernel, which has to go through a milling process, the rice kernel tend to weaken mechanically. And when the rice kernel weakens mechanically, it breaks during the milling process. That breakage is something that the producer does not want to see, because the producer is paid based on the total volume of head rice, where head rice is the unbroken rice or rice kernel that is about 75% in length of the original milled rice. And so these isotherms are very important for us. Number one, to establish the moisture relationship between the temperature and the material properties, during the drying process or during the storage process, to minimize things like fissure formation, and also, as Abass has mentioned, to minimize any potential microbial growth, because any form of microbial growth again weakens the rice kernel, makes it to fissure during any drying process. But most importantly, we also want to be able to avoid mycotoxins that could form on the rice kernel after the whole process of drying. And this mycotoxin could form on a whole kernel, or it could form on milled rice flour. And so those are some of the things that we are trying to use the vapor sorption analyzer to understand the relationships between the material, the temperature, and the moisture or the water activity within the material. Shelf-life prediction is very, very important for us, especially I'm sure you are now familiar with what we call minute rice or instant rice or individually cooked frozen rice. This is rice that has been pre-cooked, and once it's been pre-cooked, it is packed, and all the consumer need to do is to get it and put it in the microwave and then consume it. The shelf life of this kind of commodity is becoming very, very critical for the consumer. And mostly when you mix them with different ingredients, you might throw in some spices in when you're doing your processing. That changes the entire matrix of rice and throws in a completely different equation in terms of how that rice is going to interact with the moisture available and interact with the packaging material that you have. And so understanding things like the moisture characteristics in those kind of packaging environments that we are putting instant rice is very, very critical for us. And the VSA is one of those tools that is really, really helpful in terms of predicting the shelf life of different matrices of food, rice mixed with different ingredients, cooked in different styles. And so just beside understanding the phase transitions that help us control fissuring, just understand, getting idea on the isotherms that help us to predict the shelf life and also the packaging requirement is one of those things that I think we are benefiting a lot from the VSA technique of measuring isotherms.
And when that rice cracking happens, what's the economic impact? Does the farmer or the processor, do they have to get rid of that product? Do they have to use it for something else? What kind of financial impact are you seeing with these types of problems?
Well, it is huge. It's huge. Just to give you an estimate right now, when you have rice, it comes in a rough rice form. That is before you remove the hull. The price of that is about $3 to $4 dollars per bushel of rice. Now, when you mill that rice, if you get whole milled rice, then it's about $9 per bushel of rice. That is whole kernels that are just milled without any fishes. But if there occurs any fissure on that rice and rice cracks, the economic value of that rice drops to almost 60%. So from $9 per bushel, now you are talking about 40% of that is the return that you are getting. And these farmers produce several bushels of rice, thousands and thousands of bushels of rice. And so if you factor in about a 60% reduction in your income, it is huge. And of course, there's a way they are able to leverage or get some benefit out of that broken rice by taking it to maybe the animal feed industries or to the flour industries or to the pet food industries. But in general, it represents an economic loss. So any improvement that you can make on the head rice yield to the farmer really, really improves their bottom line. And that is one of the key areas that we are doing research in, preserving the head rice.
Yeah, we're talking major dollars, it sounds like, to have this issue. And I'll be interested to see your research and see how you're able to use isotherms to make some predictions, not only on shelf life, but also on the types of packaging that you should be using. Abass, I know we've been talking a lot about rice, and that's what your research is focused on, but what other products can benefit from this type of research?
Can you take that again? I'm sorry.
Yeah. I'm just curious. We've been talking a lot about rice and these specific problems for rice, but are there other grains and maybe other products that you see your research and the work of the rice processing program? Where else can this information be used to benefit different products?
All right. Thank you. So as I mentioned, we have two novel techniques we use in the lab, which are microwave heating system and infrared heating system. So we've used these two systems majorly for rice, but also we've also used it for corn and can be used for many other grains out there or any other food products that needs to be dried after harvesting, and to prevent microbial or toxin production on them. So this area of research holds huge promise, no doubt, in making our foods more safe and to limit the production of toxin in human food, pet food, and other animal feed food.
And Abass, what's next for you? Are you wrapping up some projects? Are you starting new projects? What do the next couple years for you in school look like, and what do you hope to do after school?
So in the immediate, I would say one next is to keep generating good quality data for my advisor, Dr. Atungulu here. And in the long run, for sure, I'll still be focusing on carrying out research on rice and other grains because I'm really interested in the mycotoxin part of grains. So I can easily extend that into rice, corn, wheat, sorghum, what have you. So I hope to wrap up my PhD in about 2 years from now.
And then do you plan to stay in this type of industry in grains, or do you plan to go back home? What do you think the future holds for you?
It's hard to predict the future, but for now, I'll say um, the area of research or trying to find out aflatoxin or other mycotoxin on grains holds huge promise, as I've mentioned. So there are a lot of questions to answer, and I'll be more than willing. I'm be happy to go into that and see what I find out.
And what about you, Dr. Atungulu? What's next for you? Are you going to continue focusing on this research and continue to your time at University of Arkansas? What do you see in the next couple years for yourself?
Yes, thank you. I think more than ever, I'm going to continue putting a lot of emphasis on rice-related research. Like I mentioned, rice is a primary commodity here at Arkansas. We are seeing a lot of needs. We are seeing a huge impact even in the breeding areas, which means new cultivars are coming every year with improved attributes, and those still need a lot of attention in terms of post-harvest systems engineering research. And so my emphasis is still continuing to kind of double down in that line. I want to produce more of Abass, basically by doing more of teaching and training of graduate students. And I believe when they succeed, they go out there, they become a significant resource to the rice industry. Uh, More than ever, I think I want to continue engaging my stakeholders in the field, the industry partners that I work with, so that I can identify some of the key issues that they are having and see how we can address that. So in general, I think there's a lot of opportunity for us to do more to impact the industry, and there's huge economic impact that I think we can do with our research. So that is my plan, and of course, with my role as the director of the rice processing program, I want to see how I can liaise with other faculty members who are also having interest in rice, multidisciplinary wise, and see how we can do work that uh, cuts across different spectrum. There's a lot of work that's being done right now, even in the genetics area. There are new technologies that are trying to do things like gene editing to improve the quality of rice. My goal is to find out how we can interface with them in different ways and help improve basically the quality of rice that we get.
Well, I just want to end by saying thank you guys so much for your time. I know that the rice industry, the grain industry, is really going to benefit from your research. They already have, and they continue or will continue to do so. And it makes me really happy to see people at universities like yourself using the tools that Meter makes. That's kind of why I enjoy doing my job. And we're always here to consult with you and help you get the most out of those instruments and help you lead your industry. But of course, that requires the right tools. So Thank you guys again for your time today, and good luck with the rest of your semester. And we hope to have you on sometime in the future to learn about any updates with your program and what you're working on.
Thank you. Thank you very much. We really appreciate the time to be with you.
Thank you.
You're welcome. See you guys.
Bye.
Bye-bye.
I'm Zachary Cartwright. This is Water and Food. Find this podcast on Apple iTunes, Spotify, or wherever you listen to podcasts.